hypothesis-driven experiments. Gracias.
I folded the table. See if it recognizes it in Spanish.
Thank you.
I don't know. The table internet is recognized?
Let's see, how do I know that? Do I go to the Ethernet here? Network. To be honest, I never use Ethernet, so I'm not quite sure. ¿No sabes cómo usar el Ethernet aquí en la Mac? Ay, wait.
So just another minute. Whatever you have, don't worry too much. Okay, time up. So it's okay. Yeah, just click send if you could access. Otherwise, doesn't really matter. Just think about it, and Daniela is going to start.
Okay, perfect. So, yeah, this is just an activity we're going to do every day, so you can appreciate the concepts and see if they change at all after the course, after you've been thinking about them for a while. And if you cannot send them, please don't worry. It's just more for your own, your own thinking and your own learning about these concepts. Okay, so yesterday, who can tell me what we saw yesterday? What did you learn? What, did you think about the concepts yesterday? Who can tell me something? Something about yesterday's class. Did you forget it all? Was it gone?
I remember the discussion that we have about noise in data,
Noise in data.
and that noise not always it's something bad, something is an objective of study, so this was something that I remember.
Yeah, that was a good discussion. I enjoyed that. Thank you. Someone else has something else that, remembers from Sebastian? Sebastian.
We discussed the difference between data-driven research and hypothesis-driven research. It was an important part of the class. And yes, we talked like the definition of also of data-driven research.
That's right. Thank you. It's just, I'm trying to see if you guys kind of got how it is different in concept from the more classical approach of hypothesis-driven research, and if that's clear, and then we can move forward. Yeah? Is everything good? Okay. So I was thinking that today we would see a little bit about data generation. So, of course, biology these days, a lot of it consists on analyzing loads of data, which is what we saw yesterday, and I'm sure many of you have done with analysis of big RNA-seq data, single-cell data, and all these big data sets that you can sometimes download from the internet and incorporate into your project. And of course, again, we are trying to make this course really general because we saw your backgrounds and we see that you work on many different things. So again, we're not trying to make it specific for a specific data type and tell you all about it. It's just hoping that these principles apply to all the data types that you can work with, at least in biology. But the examples, of course, that we discuss a bit more in depth may come from sequencing data, which is at least what I work in, and I think some of you also work with. So I'm hoping that that will be productive for everyone. Okay, so first, biology and big data. So big data, as we were seeing yesterday, there are many now fields that depend on big data. We mentioned some of them. We mentioned astronomy, we mentioned social media, government surveillance, these things. And there's a lot of fields and research that depends on big data. But in healthcare, we have loads of, subfields that also depend on big data. Here are some of them. I don't know if, what you guys think of big data. To me, mostly genomics, I would say, in transcriptomics. This is mostly what I see in my everyday work. But I don't know, does anyone work with, I don't know, clinical records here or more? Yeah, you do? Is this like a massive amount of data? You have like many people. So that's a different, how is it coded? For example, what are the fields that you usually see in this kind of data?
Depends of what type of answer do you want to, but in my case, we used to work with animal behavior, so we could see what neurons were firing, what type of network is working when the type of animal is doing a specific behavior, so this kind of...
And many fields, I guess. It's like massive tables.
Yes.
And tables that communicate with other tables.
And not tables, but was biological signal. So,
signals.
Yeah.
Mm-hmm. Yeah? Sorry.
I have a question. It's related to when I can call a database of big data.
That's a great question. I'll be honest, I don't think there's really a distinction. So actually, in one of the classes, I'll be honest, I can't exactly remember which one, but I think it's the success stories class. I have put several different sizes of projects. Like, for example, I think you can consider big data a single project that generates, for example, loads of single cell data. You have loads of single cells and then loads of transcript levels in each cell, right? So that is a big data set, and you need statistics and you need all these tools to analyze it. So I don't know, I don't think there is a definition of how, when it becomes big data, but when you start requiring the tools of, statistics and, inferential statistics, for example, that we'll see tomorrow, that's going to be a very dense class. I think that's when it becomes, you cannot just do a single experiment and see a clear result and, this is it. You need something else, right? You need to store your data. You need to use the tools we discussed yesterday to analyze it. So I don't think there's an easy answer for that. This is my point of view. I don't know if I don't think there is a definition.
Yeah, I just wanted to second that. I think it comes with the tools. So you might have, there is some examples where the data is just clearly huge and no one will question that is big data. And there are things that are just too small that no one, you couldn't do any statistics with it, right? You have too few data points, so no one will question it. And then you have everything in the middle, where basically it depends on your approach. If you're seeing it as big data, you're going to use the tools and the type of questions that you're going to see in this class. But you could also do another type of question that then would not be seen as big data. But yeah, I don't think there is a clear-cut definition.
Yeah, it's just, I think it became now a very used term because people are generating data at a higher speed that they can analyze it. And this is a real crisis in biology. We will also see that in the reproducibility crisis class, where people just, especially in high-income countries, let's say, at least we don't generate data that fast because it's not that easy. But some large sequencing centers just pull data out all the time. And now that's, becoming a problem in the sense of what does it all mean? Because the main researchers that generated this data, sometimes they put a paper out and they do a cursory analysis and say, but you know, who does all the, and that's when people are like, oh, you know, big data, we need statistics, we need how to integrate that. So I think that's where it comes from, but to be honest, I don't think there's like a clear, I think if you need to use statistics and, you know, have many fields, many variables connected, I think that's
You could also have,
like
there is some examples, right, Daniela, where you actually have many experiments with little data, but when you pull it together, it is at the end a big data thing, right? And I think you're going to touch a little bit of the risk of pulling together different experiments, but that's a different thing. But
get ready, guys. Tomorrow is going to be a big day. Okay. But yeah, so I don't know. I mean, I don't know if anyone wants to add. Does anyone want work with data that's not, you know, specified here? Because I'm interested. What other? There's like all kinds of data. Thales. Am I pronouncing that right? Thales.
Well, I have worked with like species distribution data, and it is a lot of data because you have many. You can like analyze many thousands of species at a time, and some species have hundreds of points of occurrence, so it's a huge amount of data.
And a temporal pattern as well. Is that?
No, it's
like
yes. The records are collected during. Of course, they're collected through time, but usually you don't take this in consideration.
For your
We just see like the picture. This is where the species is.
Thank you. So, yeah. So for example, I don't think that's there. Yeah, maybe behavior. I don't. Yeah, maybe not. But environment. Yeah, that's right. Behavior. That's right. It's interesting that. Oh, sorry. Yes. Can you remind me of your name, please?
Yeah, my name is Benjamin.
Benjamin.
So I don't directly work with it because I'm the theoretical part of this project, but in my collaboration I work with analysis of animal tracking data, tracking animal movement, and this also generates large data sets of time series of movement and things like that. It's also geographical, right? So there's a kind of coupling of these two things.
Thank you. I mean, this is interesting. I wanted to hear the data sets that you work with, because I think for the next slides, I'm going to go through a data generation process for sequencing data, because it's what I know. But I think it will apply to all, you know, all the data that you work with. And at the end, we will have a little activity just so you think about, you know, in your data set, if you already work with data, what, you know, where are the steps where the decisions that we make can change our conclusions at the end, right? So this is, okay, so yes, we're starting with biology and big data, and this is what I know, it's my field. So in sequencing, of course, as you know, there's been an explosion now of data being generated by machines and sequencing centers. And now, well, this is actually 2015, this must be, I mean, it doesn't matter which they all look the same, you know, like mass explosion. 72,000 terabytes per year in 2015. Who do you think is analyzing this data? There's not enough people to analyze this data, right? But we're not only here at this point, mostly it was genomes, exomes, and some transcriptomes. But now, for example, this single cell field is generating even more massive. I mean, from one sample, you get thousands of cells, and from each cell, you get thousands of data points. So, you know, that's exploding. That's clear. Now, there's also other big data sets in biology that I'm actually not that familiar with, but I know people work with, for example, image analysis, spatial biology, where you have a geographic dimension there as well, and, you know, as well as, you know, which cell is expressing what. So several data points, geographically spaced, and temporal patterns sometimes as well. And that's microscopy images. I don't know if someone works with that kind of data here. Yeah? It's, no, I think it's fine. And then more recently, I don't know if you've seen these massive screens. People now like doing screens, CRISPR screens, cell lines and drug screens, in which they get hundreds of cell lines and test hundreds of drugs at different concentrations, and then see, you know, the IC50 usually, you know, when the cells die. And these are also massive data sets, and to analyze them takes, you know, a lot of computational power, and of course, clinical records, as we were discussing too. And well, yeah, and what we also have talked before. Okay, So this is the point I wanted to make, and this is just a sequencing data set. Who here works with sequencing data, just to see how? Uno, dos, tres. Not that many, actually. Okay, so this will be hopefully interesting for the rest of you. But at the end, I'd like to have a little discussion about what of these applies to your data set. Okay, and this is about choices that we need to consider when we do something and generate data that can change our results dramatically downstream, which is not something that should happen, right? When one expects that your data reflects reality, but we saw yesterday that there's no raw data, right? There's always, there's always some human choices embedded into them. But okay, for those that are not familiar with sequencing data, usually this is something that you will find maybe in a textbook. And they tell you this is how sequencing data is produced. Right, you have a sample, you do some pre-processing, then there's a library preparation where you cut the DNA and add some adapters to it, and then you sequence it, and the machine produces some reads, and then you map them into the genome, and that's it. I mean, sounds very easy. And now it's usually automated. So this is why all this data is being generated, because there's pipelines. We talked yesterday that just like do this and generate it. But actually, these are all the steps, at least that I can count in my head, that happen when you are, sequencing data. So first, of course, you have your sample, and you need to isolate the DNA or the RNA that you want to sequence. And you have to fragment it, because sequencing machines cannot work with very long DNA pieces. And they cannot work, well, very recently they can work with RNA, but usually they can't work with RNA. You have to reverse transcribe that into cDNA so it can work, right? So there's a first step here where you have the DNA, you extract it, and you fragment it so it can be read by the machines. After that, you have a library preparation method that sometimes includes this step, but usually, you can select some kits, usually now there are kits, where you add some adapters to these fragments, and that's gonna allow these fragments to be usually, fixed into a plate. And then the machine can work with that, because now there's the little bit of DNA is there, and then it can read it by different methods. Then, of course, we have the sequencing itself. It can be done by many different ways. I think the most common these days is Illumina sequencing. Maybe you've heard of it. That's one of the, it's called now, it's a way of sequencing called sequencing by synthesis, where, the machine, well, there's a polymerase that is adding, it denaturalizes DNA and it adds a base on each round, and then it reads what it's adding. No? But there's many others, and there have been many other ways of sequencing DNA, pyrosequencing, for example, and there's many that I can think of right now, but this matters as well. There are many quality controls that need to be added, of course, to these steps, because sometimes the machines fail, sometimes they are not very sure of which base they added, and you need to take that also into account. Then you have to map. You have now your reads from the sequencing machine, and you need to know where they came from in the genome or that you're in, or maybe you don't know where they came from, and you need to assemble them, and you put them like overlapping each other, or map them into genome, which there are also many algorithms to map that into the genome. Then if you're interested in variants, which is in many cases why people sequence DNA at least, you have to call them, as how it's called, that you identify the variants against the reference that you map to. And there's many ways of doing that too. And quality controls again for those variants. And then finally you do some filtering and you prioritize, because obviously you're not, in many cases I would think, you're not interested in all the variants or everything that you found, but you're interested, what is associated to disease, for example, or what is the substance. This sounds, you're fine. Like you can just do it, and many algorithms do the whole thing. But I'd like now to tell you in each of these steps what can go wrong, and many things have gone wrong in the past for us as well. At some class I can tell you of all the mistakes. Actually, I have some of my own data here. Because yes, it's these things, guys. Just before we go, it's a long learning process. It's not. We can tell you these things, and we're purposely making this class for just so you get some points that are, oh, this is important, maybe I should check that. Because obviously you work with many different types of data, but also it's this experience really when working with it. In a class, you cannot really learn all of these things. Like you're aware, oh, there can be a mistake there, or maybe I should check that. But, it takes a very long time. Mariana?
And just to add, and you never stop learning,
You
right?
never.
And that's science. You never stop learning, and that's okay. When you stop learning, you are not in the right field. Just move, change your question.
Yes, no, exactly. And that's, when people say things like, well, how do I do it? What is a pipeline? I say, I wish I could tell you what is a pipeline, but it really depends on your problem and your data and your project, right? So just to say that before we delve into this. Okay, so first, the DNA and RNA isolation and fragmentation protocol. This is exactly how you break DNA before. And people used to think, at least my understanding of it, of the field a long time ago, it's like, well, you just got DNA, right? It doesn't really matter how you do it. You just cut it and put it in the machine. But, I found this paper. There are several papers like this, where people discovered that they were you follow the whole pipeline that I told you about, and then you find mutations. And then they discovered it was actually an artifact from the fragmentation. The first step, really, of the well, actually, after the isolation of the DNA, right? But and these things happen that if you isolate and if you cut DNA or fragment DNA in a specific way, and this is enzymatic I think it was the enzymatic fragmentation. Yes. Don't tell me. There's many ways of doing it. I can't remember if it was the enzymatic or the sonication. But the thing is, one of those actually causes oxidative DNA damage while you're breaking it. And that stays for the sequencing, right? And they disappear if you do it in a different method. Right? And people just didn't realize this, and people were finding mutations at the end. Oh, these mutations. And then some people were like, we don't find those mutations. This took a very long time to solve. I remember because I was at the Sanger working at the time, people were like, why are we finding these mutations and the Broad is not finding these mutations? And it was all due to this. So this is just a first step that people just thought, you cut DNA, that's it. Can introduce some biases there. That's just the first step. The second step is the library preparation protocol. There's kits to do this. So I was saying to include the the adapters into this. And there are several papers. This one uses enzymatic fragmentations as well, where people realize that some of these kits also have several steps of putting DNA together and putting it with other pieces of DNA and pulling down and all of these steps that this includes. Some of them are more sensible, for example, to GC content of the DNA. So for some of these kits, people were sequencing more often genes that were rich, for example, in AT. And for RNA, that's actually bad because you think that those are more expressed. And this is just, again, an artifact of choosing a different library preparation protocol. Of course, all these methods will have their biases, for sure. But what I want to say here is, at least you should be aware, my method actually under-sequenced GC. Maybe I can correct for that somehow, right? But at least you are aware that this can happen. And again, going back to yesterday, when you download data from the internet, many times you don't know what they did, right? So meta-analysis, again, as Andrea. Oh my God, this is. I was going to say Adriana, I'm sorry. You have to tell me a name. Again. Andrieli. Andrieli. Sorry, I want to learn all your names, but sometimes you speak to me in Portuguese and I'm like, didn't get it. Andrieli. Thank you. So as Andrieli said yesterday and made a very good point of meta-analysis. So you can download many datasets and you don't know their biases, but overall they should, you hope, tell you the same thing, right? So that's how you integrate data and kind of take into account these biases because you cannot get rid of them. They're there. But it's good to be aware of them. Sequencing method. This is like, a whole topic. Okay, so sequencing method. Right now, of course, there are, I think, I would consider, as again, Illumina sequencing the most common. Illumina sequencing has some biases that they are trying, of course, to correct with every new machine and new method that they use. I remember when I also was doing my PhD a long time ago, very long time ago, that they had these biases because Illumina sequencing sequences, let's say, a fragment of DNA by both ends, usually. You can also do single end, but now it's usually both ends. And it tended to add some bad bases that are not the ones that just in one direction of the read, not the other. Which was something really weird because no one really, like, why does it do that? Like, it makes no sense, right? And you see some mutations just in the forward reads and not in the reverse reads. And why is this? And I had Twitter fights with people about this. This is like, I remember 7 years ago, I was like, nah, this doesn't make no sense. But the thing is that this happens. I think this specific one has been corrected, but there are many biases like that, right? And this is just related to the chemistry of the Illumina machine, how it works. It has biases. So, for example, this paper here, I'm just putting examples of papers that analyze each of these steps. They are comparing sequencing methods. This one is a bit old because they use a method called 454 sequencing. This is a very old method. Maybe you've heard about. in genomic sciences, we learn about 454 sequencing. Yeah, which is a different way of sequencing and also detecting, which base has been incorporated. And they analyze here in this paper 3 different sequencing methods, and they have, for example, here 3 variants, different, A to G and G to C. The 3 different methods and 3 samples, and then they say, sometimes with a method we do find some variants, and with another method we don't find these variants, right? And it all depends on the sensibility of the method, of course. And you have a question? No, sorry. I think, oh no, this is coming later. But just to say, I know that many of you maybe are not working with sequencing data, but I think you can apply this to whatever you are doing. If there's different ways of doing something, like for example, in this case, sequencing, it's just like sequence it. That's what my advisor used to say, just sequence it. How? I need to be aware of what really, what mutations am I introducing, what biases am I introducing. And I'm going to show you some of my data later related to that. Okay, also quality controls. So anything that you do, I'm sure, when generating your data set requires some sort of quality control. And again, people just decide whatever they want to do with quality control. In my field, cancer genomics, people sometimes agree, something called the variant fraction, which is the number of reads that you have and how many, and if it's lower than a certain number, we consider it an artifact. But then some people don't agree on what that threshold is. And, people will do whatever they want, right? And for example, this is an example of a paper that I found really stunning. This is in a journal called Blood. It's a very important journal in, blood related, leukemias, for example, and lymphomas, and all these blood-related cancers. And in this paper, they studied people that have BALL type of leukemia, and people that responded and didn't respond to a certain drug called this blinatumomab. And they discovered that there was a biomarker that could predict, which people responded and which people didn't respond. And this was this thing here that is terrible. It basically means it's a gene that retained an exon, a bit of an exon, right? So remember DNA, exons, introns, spliced, got together. So it's a variant that retained an exon. And they say, the people that have this variant here, if you have it before the treatment, we see that that's like higher in the non-responders. And then the responders after the treatment, it goes high. And then they say, this is important because this gene and blah, blah, blah. Okay, whatever. And then these people basically came and said, this is an artifact of your quality control, which is, really bad. Because they basically discovered that this was, this is quite new actually, an artifact of the cDNA sequencing. So usually when you sequence RNA, You, as we were saying, you convert it to complementary DNA and then sequence it. But now you can do direct RNA sequencing. That's, to me, actually quite new. I have never used this type of sequencing, but they said, actually, if you do this, as most people do, still these days, you see that, you know, these 94 reads that are actually, you know, omitting this exon, which is what these guys found. But if we actually sequence directly the DNA, that disappears, the RNA, sorry, that disappears. It's actually just an artifact of your sequencing method and your quality controls, because, you know, they said if you had actually, yeah. Your name, sorry.
My name is Aden Yusuf.
Ale. Ale?
You can call me.
Denise, okay.
You can call me Ade.
Awesome.
I didn't understand, like I understood why this can be an artifact of the sequencing protocol, but why there's a difference between respondents and non-respondents?
To be honest, I don't understand that either. Sometimes, for this paper at least, but sometimes in cancer genetics in general, you will find some biomarkers, they call them biomarkers, and usually they have a function. They don't have to, they can just mark, you know, what is happening. But sometimes, for example, if you have a certain, let's say, a protein that metabolizes your drug, and it has different versions of this gene, because there's this alternative splicing where different exons can get together. If you have, for example, more of a version that can metabolize that, maybe you respond worse actually because you metabolize it really quickly and you need more, actually, you know, a higher dose. And, you know, maybe if you don't have that version that much, then you are, you know, you respond better because you need a lower dose. It really depends on the exact problem that you're investigating. To be honest, this one I don't know the biology of it, but usually something like that, like because there's different versions of proteins from the same gene, some of them are better at doing certain, you know, metabolizing some drugs, for example, or in the liver, yeah, doing, you know, all these things that we consume, and, you know, the active versions and all these molecules. And if you have, sometimes it's a variant in the genome, for example, that predicts you have a little bit more of this, a little bit more than that. That can be used as, oh, you know, the person that has this, maybe that means that they have a higher proportion of this version of the protein, therefore it means that they can metabolize better. Usually, I don't know if I said a lot of things that were very confusing. Was that very confusing? More or less. You are coming to lunch today, is that you? Yes, right? We can talk more about that at lunch if you want, because it's a big topic. But it's more about these, you know, biomarkers of certain, sometimes they have a function, sometimes they don't, but they're better versions for doing something, and that something is relevant to your response, for example, to a drug. And you can predict that more or less with these kinds of things, these kinds of analysis. So if these had been true, for example, you could start screening just for that. You know, you get the RNA from the blood of this person and be like, oh, you know, how much do you have? Oh, you will respond to this drug. In theory, that can be used for that. It's complicated, of course, but that's the idea. But here, of course, well, it was not true in the end. It was, but this was hard to detect at the time, because this is a very well-used still method. But, you know, some new method came and said, actually, look at the biases. Now you're finding all of these that is not true, right? Can discuss that. It wasn't very clear, but yes. More questions up until here? How much time? Oh, we still have some time. Yeah?
I do have a question.
Go, Pollo.
I'm also learning. So in this case, as you say, like a new method came, right? So there was something that the researchers could do at first to avoid this embarrassment, I guess? Or it's just part of the process? Like in this field, you do publish, and you know that sometimes it's going to be false, and that's okay, always that you did it, you know, the correct way.
No, I think that's a great question. And in this case, I wouldn't fault them precisely for not realizing, but some people said, well, you could have gone to the protein. And if, I mean, because these will code for a protein. And if you had done the proteomics, you maybe would have seen that that wasn't there. And maybe you could have been like, why is it not there? And then realize it was. I mean, I guess they could have checked that, but it's usually maybe not done.
Yeah, always. And I wonder if that is a little unfair, right? Because it's like,
oh,
do unfair. A whole other paper to test your paper.
Yeah.
And there's, I mean,
True, although here they are saying these people respond to this drug. I mean, when you're making claims that are directly relevant to the clinic, I think you have to go further.
Fair point, fair point.
It's hard, it's hard. I think
They should say this suggests this other research will be needed to confirm.
Yeah, I guess.
And
exactly.
we are not doing it because it's either outside the expertise of our lab or because we just want to publish and not do another 4 years.
I do agree with that. Another thing? No? Okay. Now, let's go to the next thing, which is the mapping algorithm. So far, what have we done in this sequencing data set? We have extracted DNA, we have cut the DNA, made a library, and sequenced it. And now we have reads. The algorithm just, gets reads out, short reads, and then you're like, whoa, what is this? Where did they come from in the genome? We have to do that here. Okay, so for the experts, this is data from Peter Danicek, who is a researcher at the Sanger Institute. And this is how a mapping graph will look like for people that have not seen this graph. Here you have the reference genome. This is like your reference, like this is what you're mapping to. And these are annotations of usually if you see a boxes like this, that's a protein coding gene, an exon. So because you have a Q, G, T, whatever, right? So this is what these three bases are coding for. And then here you have DNA. And when you see this is the mapping, right? The algorithm says this fragment of DNA belongs here because, it's the same sequence. So I say it's here. And if you see a letter, actually, if it's gray, it means it perfectly matches what's here. But if you see a letter, that means there's a mismatch, so a mutation. So in this experiment, in DNA, you will always see some. These are errors from the sequencing machines. For a real variant to be there, you need to see, a straight line or half of them in the case that you're diploid. But here, when you get the DNA from the same, this is the same sample, right? You have DNA and you have RNA, 2 replicates. And here, well, there's nothing, right? But the RNA samples seem to have lots of variants. For the experts here, who can tell me what's happening here? It's a mapping artifact. Anyone that works with this data? Any ideas? The question is, why do you think that when I get DNA and RNA from the same sample and map it to the genome, I see no mutations or no variants in the DNA, but I see it in the RNA?
To take some wild guesses. So
Yeah.
don't get
I know many of you are not familiar with this data set. Both. You extracted both.
Repeat the question so they hear it.
Anyway. I repeat the question? Sure.
You just answer.
I'm confused.
I just asked if the original sample was RNA or DNA, and
both. You extracted both DNA and RNA from the same sample. So why would a mutation appear in RNA, or what does that mean?
I would say that the replication of DNA. Well, I do not work with that. The replication of DNA, conservation of what you're replicating is far more important than during transcription. So I think transcription may have more misplacements.
RNA editing.
What?
RNA editing. There is a method in which a mechanism in biology where mutations are added to RNA. Right? That could be. I mean, that's a good guess because that happens. You have you?
I don't know if it's something related to base deamination.
That could be as well. It's a mechanism of RNA editing, right? Yes. So this is all true. Yes. The problem here, of course, RNA editing, and we'll see a paper about that actually. Now that I remember, usually happens from A to I. I is an intermediate base. It doesn't happen with all the bases, and you see here some C's and T's and G's. And so in this case, what's actually really happening is a problem with the mapping algorithm. you have in RNA a process, so you have a processed gene. Remember, for the non-biologists here, again, when you have DNA and this is a gene, what we were discussing, what is a gene? Let's assume a gene. Then it gets transcribed, and usually if it's a protein coding gene, it will have exons and introns. So the introns will get spliced out, and then the exons will get together, and that, you know. When you sequence RNA, you're just seeing the process, the final, usually, you see everything. But in this case, you're seeing the final, you have cut this bit out, right? This is an intron. This is exactly the boundary between your exon and your intron, right? So in this case, someone used for this a mapping algorithm that's called not splice aware, right? So it will not know that it needs to add a cut here and map these bits to the next exon, right? So this is actually, people would say sometimes, oh, there's mutations here. This is not a mutation. This is just a problem of the algorithm you selected for mapping. Is that clear? For at least, you know, I told you this was going to be heavily genomics based because that's what I know. But I'm hoping that you kind of understand the problems here with this.
To add something here. So the two ideas that were good ideas were focused actually on the biological part of it, right? Or even like the generation of the data. And this is actually a problem of the processing of the data. So just to keep in mind that part, that it could happen at several steps, and all of them are important to check again.
Exactly, and because that exists, that process exists, there is a paper that we will discuss that they found a lot of these things, and they claimed, you know, we found a lot of these new methods of RNA editing, and it was all artifacts. And yeah, this was a big paper at the time. We will see it in one of the classes. But yes, so you have to be aware of that. Here, this is my own data. I know it looks ugly. I know we have a class on visualization. Do not do this. This is just working data. I do not intend to present this in a paper, but I like to just show you some examples here. Once we have the DNA mapped, usually, well, not usually, people do many things with it, but us in disease-related genomics want to find mutations and variants associated to disease. So we have to do the variant calling. You know, where are the variants? And there are many methods to do that. There are different kinds of them. There are Bayesian methods, there are heuristic methods, many different kinds of methods that you can use. And here I use 3. At the time, very popular. This is a bit of old data, so actually this thing doesn't exist anymore. Good, because it was really bad. But you can see, we called variants with these 3 algorithms, and only 68% of them actually overlap between 3 methods. Some of them were just called by one of these. And this is just another way of seeing this data here. This is just a genome position. All the variants that I was detecting in samples, we have like 6,000 samples. And if it's green, it just means that that variant was detected in that sample by 1 algorithm. So green in this case just means 1. And if it's darker, that means it's, you know, detected by black is all of them. So this is a real variant. You could say, okay, you know, this is detected by many. But many of these variants are just detected by 1. And in many cases, people just use 1 method because, you know, that's the one that is installed. That's what this paper did, and they just used 1 method. And this is heavily biased, actually. So at least us in our research, we try to use at least 3 and try to take the intersection of that because we think that adds, because it's different approaches, we think that adds to the probability that is a real finding and not, you know, an artifact. Is that clear, more or less? Okay, now quality controls. This is also a figure from my thesis a very long time ago. But basically, also in variants, when you detect variants, that also is biased. That also has qualities, many quality metrics associated to them, because there's many biases that the sequencing can introduce, and the mapping, and, you know, and all these steps. So usually, variant calling methodologies add something called a quality score. And here, what we did was trying to confirm these variants by a different method. So I was saying, right? And if the quality score, obviously, yes, was low, that meant that the fraction of confirmed variants, obviously, was lower. But to know where to make the cut, That's the problem, right? When you say 20, many papers use 20, and out of the 20, 80% are not, real. You see many papers, oh, well, quality 20. You're like, well, actually, quality 20 is quite bad, at least for these data set. Again, this changes, right? You have to make these tests for your data when you do this. And if possible, it's not always possible, but if you can, it's always good to test whatever you think your result is by a different method, if it's possible. And these are capillary traces. It's a different way of sequencing, which is a different chemistry, different ways. But basically, if you see these little mountains, that means bases. And if you see this, I have 2 here, that means I have 2 bases. You have 2, you're diploid, so you can have 2 molecules of DNA going in. So that means, oh, this is a real variant because I see 2. It's heterozygous. And in this case, I thought there was a variant, but in this place, actually, I don't see anything. So that's not a real variant. Right? We're about to finish, guys. Don't worry. This is our last slide, and this is scientific gossip. Who has heard this? Who has heard about this paper? I know it's old. It's like 2014. You were very little, but it was a big scandal. Anyone has heard of this paper? Stop. No. OK. Basically, and this is something to do with the filtering methodology or the quality control. I mean, this is all together. In this paper, that was published in 2014. This was a big, big paper because it told the world that you could create pluripotent cells by putting normal cells, like normal, I think it was mouse blood cells, in acid. In acid. Like you put them in acid and they became pluripotent. And then they did more experiments with them. Then they put them in some mice and got them pregnant, and then they saw, these cells actually form placenta. And pluripotent cells actually cannot form placenta, as far as, they have a limit. So then everyone was like, oh my God, like you just put them in acid and we have all these, it's so easy. And this was 2 papers in Nature actually at the same time. And it was a big, big scandal. Like people were like, well, we have to do it and this is so easy. Because before this, and still, you have something to say? No? Before this, and still to this day, because this was fake, people had to use these Yamanaka factors. I don't know if you've heard that. You add 4 and then, you make how to say this, transdifferentiation. I think it's dedifferentiation. And then back to the states and, But these guys were like, none of that. Put them in acid, that's it. Basically, that was the paper. And, time is up, sorry. We're about to finish. And the problem was, then of course, some people came and said, well, actually, this was contamination. So someone had given these guys some cell lines that were normal ES, what is ES? Embryonic stem cells from mice. And these other guys had done a lot of things to them and put them in acid and I don't know what. And in the end, these other guys reanalyzed the results and said, actually, there's 2 problems here. The first one is that you told me that the donor of these cells for your experiments in the mice was a female, I think, and when we sequence this is actually a male to begin with. Like, this is not the cell that you're telling me. And the second one is that you told me that the cells were pluripotent because you could detect with fluorescence this OCT4 factor. It's one of the Yamanaka factors. And when cells express OCT4, it's a gene, it's a marker, as we were saying, a biomarker of pluripotency. But they were green, but some other researchers in this paper say, if you know the data you're working with, and if you know this field, you know that stressed cells will just glow green, because that's how they detect them by fluorescence, because they're just stressed and they're expressing loads of things. And if you detect that, that's just a low-level baseline, and these guys just didn't know that and published this massive thing. And then it was actually a tragic story because some people committed suicide here. Because it's Japan, and in Japan it's very, it's very strong. But you can search for this. It's actually a whole Wikipedia article about this scandal. It's it's a big scandal, and you can look it up. But this was all to do with basically not considering your contamination in your cells, not checking properly what these things were, contamination and your baseline levels of And then some people said there was also some fraud, but that I don't think that's completely been proven. But okay, so just to summarize, yeah.
I just wanted to add something, like more a general advice that I heard multiple times during my PhD, and I think you're hopefully also going to learn multiple times during your career. Big discoveries need big proofs. So this thing, like if you find something that is going completely against what all the previous research does, double, triple, 4 times check. That is the time that you actually have to go through very much details. If this wasn't like a big deal, there was probably a good chance that 1 is correct because it's in line with everything else you know. Second, no one will care because there is also, and we are going to talk about that, there is also a lot of false science or just things done wrong published, and no one will make a big deal out of it. That's how science goes in general nowadays, at least. But if you have something really big that is going against, or it's just going to have big implications, I guess the other thing you say, right, Daniela, with if it is going to patients right away, or it's going to change policy in a way, then you have to have big proof for everything to do. Yeah, that's it.
No, I agree. I agree. I mean, it's just, I wanted to mention just one thing that went wrong at all of these steps, that is sequencing. When someone tells you, I sequenced my sample, they did all of that. But, what exactly did they do and where are the biases is something that many times we don't know. And if we're doing it, we want to be aware of them. You cannot remove the biases because all methodologies have biases. But you can try and control for that and at least be aware, as Mariana is saying, if I find something that is like, I put a cell in acid and it becomes, that's like, it probably doesn't agree with the body of literature that has been published, right? So you have to try and test in many different ways your observations, right? That's just all we wanted to cover here. And now I just wanted, maybe a minute, but I've told you about sequencing and this is what I do and what our lab does, but what data generation steps can have, an impact in what the steps that you use to generate your data in whatever field that you're working in, and how do they affect, how can they affect the conclusions of your study? And this is just something that I would like, of course, because I'm not an expert in all these areas, it's just something I would like everyone to take from this class. Like, just think about the steps and think about where the biases could be, and so that you're aware if, maybe you have a false positive or not at the end of your study. Okay, so here we conclude what we saw, and now we have an activity, but okay, the summary for today, it's that you have to be careful and conscious of all the choices, because at every step, if you're generating your data, you're making choices. I'm going to use this library, and I'm going to use this sequencing set, I'm going to use this variant caller. So just be conscious of your choices, have a clear idea of why you're choosing that, not something like, it's already installed there. I used to do it too. One learns, I tell you, it's like 10 years, or I don't know how many years I've been doing this kind of thing. But it's like, okay, fine, it's installed, but why is it the best method? Or what biases does it have, right? Just be conscious of that. Try to assess, if possible, the accuracy of every analysis step. Where are you introducing these false positives? If possible, at least in variant calling and in mutation research, in many cases, we can test our final results against the original source. So sometimes you have more data, more tissue from the original patient, for example, and you go again and say, is this mutation there? Because it went through all these steps. Is it a false positive, or is it actually there? So try to test that. And if you're considering, of course, downloading data, which is good, I think we should use it, but just be sure to read the papers where this data was generated and know which choices the authors took. And if you're in doubt, you can probably contact the authors. And in many cases, they will reply. Some other cases, they don't, but you can try and decide if you use their data or not. And that's it. Now we have an exercise. I think Mariana will tell you about the exercise.
So yeah, the exercise is actually conceptually really simple. It's just the idea that, Daniela has explained you a lot of things that you need to consider when you are working with data or just producing the data. But there is something like behind it and like a little more like conceptual, and it comes to the motivation. Why are you generating data in the first place? And some people, particularly in this reference that I'm putting here, separate the motivation in three. It could be done just as convenience experiments, and that's something important that we don't always think about. To be honest, for me, I didn't really consider this option. Like if you will ask me before we were planning this class, I will only say exploratory and hypothesis-driven, but actually many experiments are done, and I would say experiments, models, research in general, is done actually because you already have the option there. You have the tools, you have the setup ready in your lab, and you say, why not, right? So that will be convenience experiments. We also have exploratory experiments. I think that is more connected to what we think traditionally as data-driven research. You want to know what is happening there. You still don't have a well-formed hypothesis, and you're seeing just what you get out of it, right? And then hypothesis-driven experiments, that is the more classical picture, where you actually have a very, very clear hypothesis of what might be happening, and your experiment is done to test directly that hypothesis, right? So those will be the 3 points here, very general, but I actually want you to go in groups, and the same groups that last class and the rest of the course, and discuss what are the implications, what is the things that we need to think about, what are the potential pitfalls, what could be the benefits of these 3 different types of experiments or way of gathering data. And you actually have 15 minutes, so enjoy, have fun, thinking. Are also like these 3 things clear cut, or maybe there is something in the middle? And then you have to summarize the main points in your discussion, and in the last 10 minutes of the class, we will share it with the rest of the group. Does that sound good? Any questions? Okay. I will count the time and let you know as time progresses. 2 more minutes, so start thinking about main points to present to the rest of the group. ¿Va? Sorry. Okay, guys. I should count as well. Okay, so let's start the discussion as a group. So I don't remember the teams, but if you know or we can go voluntarily. So which team wants to start saying the main points of their discussion? Anyone from the team? Any team?
Okay. No. Can I stay here? Okay, because I'm going to read what we tried to summarize. Okay. No. Hi guys. I'm going to read what we as a group tried to summarize as our ideas. So for the convenience experiments, we thought that the implications could be limited conditions and restricted to more convenient resources. So maybe we could have unusable data flooding. So we would generate a lot of data that is not. Now, what are we going to use it for? So useless. Potential pitfalls of these kind of experiments would be wrong or biased conclusions and spurious correlations, because we have so many data, random data, kind of this kind of thinking. And benefits could be that as you already have the mechanisms, the machines, or these kind of things to do these kind of experiments, these could not require new resources, and it would be low cost and probably fast, because you have all the things you need to do it. Concerning the exploratory experiments, we didn't exactly wrote implications, but we pointed out that potential pitfalls could be also spurious correlations, because you're just looking for, what can I find? So we're going to look for strange things, maybe. And benefits would be that we can generate a posteriori hypothesis, because as you're looking without having something in mind, we could reach new things that maybe in the field are not taught yet. But yes, as you're looking without a theoretical bias, you can find this kind of new hypotheses. And the last one, for the hypothesis-driven experiments, the implications could be that sometimes it would require specific tools and methods, because you need to now you have something that you want to achieve, a goal, a hypothesis to prove or disprove. Potential pitfall would be that it requires previous premises. That before starting these kind of experiments, you must have a clear hypothesis, and it would take more time to design an experiment to avoid bias, because in these kind of experiments, you would be more tempted to force a certain result. So it's harder for the scientist to eliminate himself or herself of the experiment. Benefits of it could be that we are discussing that this kind of experiment could provide a more robust conclusion, because, yeah, yes.
Okay, so it's working. Yeah, perfect, thank you. That was really well summarized and put. Another team who wants to share?
Yeah, I wasn't talking with the professor that like the convenience experiments could be both inside the, okay. The explanatory experiments and the hypothesis-driven experiments, in a sense that we kind of have two types of convenience experiments. One in which you are out of money, and then you have the tools, you use the tools that you already have in your lab, and then to do these experiments, they could be also explanatory and hypothesis-driven. You know, it will stay being a convenience experiment. And the other, that is when you have a plan of money, and then you don't need even a question. You just do it because you generate some data. And then you have the problem that you're generating a lot of data that you don't know if will be analyzed, if you know how to analyze it, and if will actually turn into knowledge someday. So, yeah, I believe this is.
Perfect, thank you so much. Another team?
No.
Well, I want to comment something, because well, now I understand that we are talking about experiments making in a laboratory, but also we have data in, for example, for an epidemic. So in that cases, for example, in COVID, we didn't know anything about that illness. So it would be necessary to make all of possible that we can. So in that cases, I think that exploratory experiments, I don't know if it's called, well called exploratory experiments that, well, it's necessary to have all the information that we can.
So, but don't you think that's a really good point, right? But don't you think specifically in COVID there is as well a part of convenience experiment? When we decide to use, like for instance, Google information for transportation, is really Google the best option, or is because it's the convenient option? Or how they intersect with each other? But that's a really good point. That's a really good point. It's not always an experiment that you decide to do. Sometimes it's data that you are collecting, and it's exactly the same kind of process, right? You can also collect data because of convenience, because of exploratory, or because of hypothesis-driven experiments. And you are not seeing this in this class because it's kind of different, but it's the kind of things I do. You can also generate data from mathematically, from a model, and you still are gonna have these kind of issues to think about. But yeah, that's a really good point, thank you. Anyone else, another team in particular want to share their main points? No? There has been 3, so.
We discussed a little bit about the probability of getting results, and that was one of the benefits of the hypothesis-driven experiments, because even if we don't get a result, it is still a result. Whereas in the exploratory experiments, we can search for a lot of things, and there is always a probability of not finding anything. And we also, it was interesting because they put like a lot of, if I correct, you guys talked about in the hypothesis-driven that you need a lot of direction to do what is hypothesis-driven, and you put it as a pitfall. And we thought about that as a benefit, because when you have like a clear direction, you can again get to better results as well.
Just to mention that what you were saying about like hypothesis driven, if your answer is negative, you still have a clear answer versus the other ones. That's a really good point. Really good point. Thank you.
We can assume that every data comes from an experiment because, for example, in COVID, we are changing the phenomenon. So we are not. I don't know if it is an experiment, the data from COVID, for example. I mean, like the cases and deaths, the data that we use to create models and everything. This could be an experiment or not. I don't feel that this is an experiment.
Yeah, that's a good point. I think it kind of lies on semantic, and maybe Daniela has another point of view, right? But just in semantics, what we call an experiment. And I think it's very, it's open, right? As I say, I work with mathematical modeling. I actually produce my own data, and we still call that an experiment, right? Because you are kind of you're making an experimental design under which you are going to be producing. You have a set of assumptions, you have a specific way you're going to test that, and you get something out of it. Now, mine is very hypothesis driven, so it might be different for the other ones. But I think you have a good point, and my take on it is it really doesn't matter when you stop calling it experiment or when you start calling it experiment. What is more important is what you're doing with it. So for me, in the moment you are testing something, and I guess we always say testing in terms of hypothesis, but you sometimes are testing for patterns, or you are testing for information. For me, that would be an experiment, but other people might have other takes on that.
Remember yesterday we also talked about observational research. So sometimes you do an experiment and collect data, but sometimes you're just observing things and collecting data from that, but you're not interfering directly. I guess when you collect data, maybe you are interfering in some way. We can also discuss that, but I see it more, that's my point of view, but again, yes, it's open and people can see it in different ways. It's a form of that kind of, usually the people use it for like observing species in, their environments, but I think here you're observing a phenomenon and collecting data from it, and that's another way of, also getting data, but also it could have some, choices and experiments and then play a role, right, in the results that you will get from that. Thank you, that's a good point.
So anyone else wants to add something? It can be a short comment, yeah.
Yeah, our group, there's a point that everything's like overlaps, but we have another point that we haven't talked about. That's in the hypothesis driven, it can lead to like a pet hypothesis, and you can like design your experiment around proving your hypothesis. So this would be like one of the pitfalls of hypothesis driven, and it's discussed this in length in our group.
Mm-hmm. You're already kind of looking for something, and I think Jay was kind of mentioned, it can bias the way you think about stuff. But as you're going to see a lot through the lectures from Daniela, like data-driven or exploratory research is not exempt of bias, right? And I think that's in general true for science. We have to be aware that we all have biases that come from different sources, and yeah, you just keep them in check. Never assume that you don't have them, because you all have them. We all have them. It could be for our background culturally, or even our background academically, or just our desires of seeing something. But yeah, that's a good point in general. Okay, so we are running out of time, so I think we are going to stop there.
I just want to say something. So this little experiment and discussion, it was just more, and this has come out in the discussion, just to see when you generate data and when you want to find out what it's telling you, it does matter, how the experiment was designed, of course. So sometimes you can only detect correlations, but if that's, you generated data just like that, and then you see some correlations, that could be spurious, like, you know. But if you design the experiment because you already knew, you had some idea that that could happen, and that shows these correlations, that maybe, depending on how you designed it, then that can be confirmatory, right? So it's just, you know, it's important how you design the experiment, of course, and how, you know, you got your data. And this is, you know, semantics, or like people call these ways of doing things differently, but it's important to know them when you're designing your experiment, or you are, you know, downloading data and trying to decide, you know, how to find answers for your questions, right? So it was just to conclude that. And I think, yes, just very quickly, can you fill in again? They are the same questions. And hopefully you have internet now.
Yeah, just as before, I'm going to close in 5 minutes. It is already in Slack. It's also here. Just the best you can answer. We're actually just pulling all the answers together. It's not to evaluate each of you individually. It's just an interesting process to see how these conceptions change as the lecture goes. So just please try to answer briefly. And yeah, as soon as you're done with this, you can leave and have a good rest.
And we'll see you tomorrow.
Because tomorrow is heavy.
For statistics tomorrow. Difficult topic.
If it's to see how the lecture goes, would you say it's
Time up. If you haven't clicked send, click send. I'm gonna close it. Yeah.